So tadpoles. Why do we look at tadpoles in relation to chestnut trees? And especially in the Northeast, I don't know to what extent here, but vernal pools are kind of a really unique ecosystem component of deciduous forests. And these are temporary ponds that tend to fill in the spring, and many different amphibians, especially salamanders, also frogs, snails, various species, use these temporary ponds. Amphibian eggs hatch and start to develop in the ponds. Ponds usually dry up by midsummer and then kind of fill again the next spring. So vernal pools are unique ecosystems, kind of indicators of critical and good habitats. And we want to make sure that, so these pools are lined with deciduous leaves, typically. And in the era of chestnuts, they would have been lined commonly with chestnut leaves. And so if we were able to restore chestnuts, chestnut leaves will be in pools and exposed to tadpoles. So we, to work on this, got out hundreds of quart ball jars and put one wood frog tadpole in each jar. And then we had 6 different leaf types. We looked at sugar maple and American beech as trees that are already out there in the ecosystem. Chinese chestnut and hybrid American Chinese chestnut as controls for what could be out there, what's being planted. Not so much in the woods, but they're out there, they're not regulated, there are comparisons that people are kind of talking about for potential restoration. Then we have non-transgenic American chestnut and transgenic American chestnut, and then no-leaf controls. And then for each of those, we had some that had supplemental tadpole food and some without any supplemental food, these are just plain leaves. Of course, the no-leaf controls, we only had the supplemental food. We needed to give the tadpoles something. We didn't have a plain water control. So, what did we see? Oh, here's just a little bit more detail. Here's a tadpole in one of the jars. You can see some of the kind of crushed up dried leaves in the jar. Here's looking down on a bunch of jars. And here's our whole study set up with 195 jars. And this took a lot of time this spring. And here is, I think I might need to start it on the computer, just a few seconds of video showing these, did that start? I just wanted to show that, that the tadpoles actually, oh come on, ate the leaves. I think we're going. No? Ah, no video. Bummer. Okay, well, you imagine the video, there's a tadpole swimming around and it's actually eating leaves, so.
You got it.
So, we also took pictures and, all along the way. This is pictures of the same tadpole from the same jar. So we have this graph paper in the background, and so this will let us actually use software later to scale how big the tadpole was at each of these. We can look at size later. And then you can see by the end they're developing front legs. At that point we had considered them metamorphosed and we released them. Some data looking at overall survivals sorted by leaf type. The transgenic American chestnut was a little lower, 80% of them survived compared to 90% of the non-transgenic controls. But that's better than the Chinese chestnut, sugar maple, and American beech controls. So we're not making, if this is a real change and not kind of a fluke from our sample size, it's not a change that's beyond what's already out there in the woods. And then interestingly, the no-leaf controls, 80% of them died. The supplemental food alone was not enough to sustain the tadpoles. Looking at that in a little more detail, this is developmental stage. So there's, I learned as a chestnut researcher, didn't really know this about amphibians, but there's this scale of tadpole development, starting from just hatched and ending with front legs and basically a frog with a tail, called the Gosner stage. So we each week evaluated what stage every tadpole was at and got these averages. And after 3 weeks, here they are sorted by leaf type again. The kind of lighter colored bars in each one are the ones that did not get any supplemental food. And the darker ones are the ones that had both leaves and supplemental food. So one of the most distinct differences we saw was that as long as it had some leaves and some supplemental food, the tadpoles developed. for all of them, and none of the differences were statistically significant. The ones that didn't get any supplemental food developed slower, all across the board. And then the ones that didn't have any leaves, that only had the supplemental food, 3 weeks in were kind of in between, but survival was a lot lower for those two. So that was one of the biggest differences. Leaf type didn't matter so much as whether it had both supplemental food and leaves. Then looking at the ones that, this is only the ones that did not get supplemental food. So everything that got supplemental food after 8 weeks had basically developed, and we released them and they all developed, pretty much. They all survived, pretty much. These are only looking at the ones that did not get supplemental food. So it might be more representative of what's going on in a vernal pool. We're not quite sure. This is a pretty artificial setup here with jars in the lab. But interestingly, the ones that got American chestnut leaves developed more than everything else. It didn't really matter whether it had a transgene, but American chestnuts seemed to be providing something that allowed tadpole development more than other leaf types. Even hybrid chestnut leaf types. So that's an American Chinese hybrid, and the full American chestnuts allowed for better tadpole development. Pretty interesting. American beech in this case was lowest in terms of development, but also survival. And the ones without the supplement, many of the ones with the beech leaves died for some reason.
I've got a question for you. I'm organic garden, and I've got a chart that has the different acidity of leaves, and all of them are acidic with the exception of ash, which is in a neutral pH. Did y'all see any difference on that? I wonder, is pH also playing into this?
We did check pH on these. I don't have the data here, but there were
I'm
not...
sure it's acidic.
Right, they're all acidic.
Yeah.
There were not major differences between them, especially between the chestnuts. So if pH were the only factor, then the hybrid and Chinese chestnut would have come off the same instead. So, enough about the tadpole studies. I'm going to kind of get back into some more general stuff about how do we, so I guess assuming these things are safe and they're not detrimental to tadpoles, or anything else in the environment, or our diets and so on. And if you want to proceed with creating a transgenic plant, how would you do that? We use a process called agrobacterium transformation, and that's taking advantage of a bacteria that naturally lives in the soil. And this bacterium puts a little bit of its own DNA into a plant. Here's kind of the schematic of what happens. There's a bacteria in the soil. Here's a little bit of the bacteria DNA. This little chunk, the one chunk, gets transferred into a plant cell. And in nature, this creates a gall. Agrobacterium makes a gall, and it kind of creates a habitat for itself. 40 or so years ago, some really smart scientists realized that we could replace that little chunk of DNA that the bacteria puts into the plant. We can put in any gene we want right there. And the bacteria puts it into plant cells. So, and that seemingly low-tech solution is still one of the best ways to move good-sized chunks of DNA into a plant. There are newer technologies that are really good at precisely moving small chunks of DNA, but for moving whole genes or multiple genes, agrobacterium is still about as good as it gets. So, we can use the bacteria to put a gene of our choice into a plant cell, but it only goes into one cell. I mean, it can go into multiple cells, but if we were to just kind of expose it to the whole plant, there would just be kind of cells here and there that have taken the new gene. So, to make a transgenic plant, we need to grow a whole plant from one plant cell. And that is a bit of a trick, but not impossible. This is our whole process. And I'm going to go through that and then come back to the transformation, to the putting genes in part. We start on chestnuts from immature burrs. So we collect the burrs mid-summer before they are actually ripe, and then we slice the little immature nut in half, and we can get these embryos, they're called, eventually called somatic embryos. We get these embryos, and we can put those onto special kind of jello-like medium and culture them. We can promote them, or we can promote continued growth of embryos. And they kind of grow and bleb off and kind of keep making new embryos. And we can keep them at this embryo stage and keep making more and more and more embryos and split them apart, and they keep growing as embryos. until we change hormone concentrations. We can kind of stimulate them to grow into these little green blobs, and then the green blobs eventually turn into something that almost looks like a plant shoot, and from there we can put them into these cubes about this big, and so the little shoots are about this big, and we can keep producing more and more of these shoots that all came from a single embryo, and we can basically in the lab make as many as we want eventually. So we get this little forest in a cube, and then again, changing the conditions, we can stimulate them to grow roots and eventually produce a whole tree. Now the stage where we put in the new gene is at this embryo stage. So here's new gene or trait. And so when we expose these embryos to the agrobacterium that moves DNA, the DNA will only move into one of these little one cell on one of the embryos. So we need to make sure that cell stays alive and grows into a whole embryo derived from that one cell. And then that embryo can develop into more embryos, and then we can stimulate this development to a whole plant that came from a single transformed embryo cell. So that's kind of the next step if you'd be thinking about Ozark Chinquapin transformation, is getting Chinquapin burrs, probably right now, or maybe about 2 weeks ago, but sometime right, kind of midsummer.
We'll send you a variety of eggs.
Yep. So, and then trying to isolate these embryos and to be able to culture the embryos. And once we can have this kind of sustaining culture of embryos, then we can do transformations. We can expose them to bacteria, put in the new DNA, and then start the rest of the process. With chestnut, this one slide is about 20 years of research. So hopefully, now that we are familiar with most of these, it'll be a matter of minor tweaking to get it to work with Ozark Chinquapin and not starting from scratch. It's a pretty science-heavy slide. Any questions? Anything else I can clarify? All right.
Incredible amount of work that went into doing what you're doing, checking, a lot of different avenues of effects and all. It's pretty impressive.
But that whole tissue culture process that I just described, so we get these kind of funny looking shoots and we can plant them and they survive, they are okay outside, but they tend to grow slower. And you can imagine here the nut, most of the nut is just stored energy and nutrients for the new plant. And so, not surprisingly, when we plant a nut, a seedling, it grows a lot faster the first year. And if these survive, they can catch up after a couple of years and start to grow at about the same rate. But anything in a seedlings, anything that comes from a nut is going to have a head start compared to something that came from tissue culture. So there's a couple of kind of important notes there. For any sort of distribution, if you're giving these things to other people to grow, you want to give them a seedling. You don't really want to give them this little kind of slow-growing, hard-to-care-for tissue culture shoot. But also, everything produced in tissue culture, remember I said it all came from that one cell? It's all genetically identical. They're all clonal. Which is great for testing and production and it's consistent, but not so good for restoration. You don't want a genetically identical forest. So, if we can, or when we can grow these tissue culture plants, get them maybe this tall, we can, I think I go into this in the next slide a little bit. We can grow them in a growth chamber, real high light level growth chamber. We can actually get them to flower in about a year under kind of optimal conditions with high light in a growth chamber. These are catkins in a growth chamber. So we can collect the pollen from a transgenic plant, collect it inside, take it outside, and do pollinations. There are a number of conditions we have to maintain with our permits in order to do this, but we can do this on permitted test plots. We do the pollinations, and at this point you have to remember a little bit of basic genetics, that chromosomes come in pairs, and when we insert the gene, we only insert one copy. So it only goes into one point on a chromosome. So that means that the offspring, about half are going to inherit the transgene. So if you remember back to Punnett squares, maybe from genetics class, the important point is that about half of the offspring inherit the transgene, and about half don't. The ones that do inherit the transgene are fully resistant. They're just as resistant as the transgenic parent. The ones that don't inherit the transgene, they're normal American chestnuts, not regulated, totally blight susceptible. So offspring, about half get it, half don't, but it's easy to track, and if they do inherit, it's fully resistant. So that's kind of a contrast to the breeding program of Chinese chestnut, where there are several different genes that control blight resistance in Chinese chestnut. So offspring might get kind of different complements of those, and you might get some offspring that have a little bit, but not all of blight resistance, and it's inheritance is very complicated when you get the trait with several, controlled by several genes like that. Because this is a single gene, one chromosome, the inheritance is pretty simple and pretty predictable. And then you get chestnuts. So, thinking kind of longer term on this, the next step we would want to do for a larger scale experiment, and we have these envisioned a long-term ecological test type test plot here. And we have kind of restoration forests where transgenic chestnuts would be planted along with other native plants and maybe blueberries and vernal pools, and kind of make a realistic type forest. And then we also have some more orchard type plantings that we could observe. But then, so if this were our transgenic plot, we're testing transgenic chestnuts, we'd also have a plot for non-transgenic chestnuts, and we'd hope to keep them alive with hypovirulence probably, and then a plot for hybrid chestnuts or backcross chestnuts. So again, we could compare any effects that we're seeing kind of longer term on the more landscape ecological scale as opposed to our small permitted test plots.